could read the nucleotide like A, G, C, T like that. So for that accuracy, they have mutated it. And the first instrument they have released, that is Minion. So this is a small equipment. Suppose if you have a ABI a sequencing machine, you know what is the size of that and how much machinery is there and software is electronic equipment is there inside it like that. And there will be a robotic hand also will be there to handle the samples. So this is a very simple machine. Suppose if you have a Illumina or ABI, there is a high investment cost is there to purchase the equipment. But here, if for the nanopore sequencing, the equipment purchase is very low. You can purchase this Minion for around 2.5 lakhs for starter pack. So this is the third generation. The Sanger method, what we told is first Maxam Gilbert, the kernel first generation, Illumina second generation, PacBio and nanopore technology. That's another third generation sequencing method. So this is how the MSPA is arranged in the memory. So this is the MSPA protein which is there in the non-conductive memory. So the DNA will be directed to the pores through adapters and the motor protein like so the motor protein will pass the single strand through these pores until the end. Then in the earlier first generated flow cells, after passing this first strand through the pores, the second strand also will be passing through the pores, so that a double-stranded DNA is sequenced in a single pore. But later they have modified it in such a way that only one strand will pass through it and it will be sequenced. Because both strands, when it pass through it, it is the amount of data generated may be from the same fragment several times. So they have made it the adapter ligation, they did it in such a way that only a single strand is passing through the pores. And the other strand also will be passing through it when the adapter binds to the other 5' end. So you can have from different fragments, you can get more number of reads. So that is how sequencing event is taking place through the nanopores process. So they have made it in such. So when you see these pores, this is the DNA spanning region in the pore. So there are 6 nucleotides or 5 nucleotides like that. It will be passing through it. So the electrical current generated for these combinations of 5 or 6 nucleotides. So in the case of Sanger's method or in the ABI machine, when you see, you are using 4 different fluorescent dyes. So based on the 4 different fluorescent dyes, when the laser falls on that, based on the 4 different colors, it is doing the base calling. But here, when the DNA is passing through the pores, the membrane is spanning 5 or 6 nucleotides in it, and when the 5 nucleotide passing through this, for this combination, a current will be generated. And when the T enters into this, then TGG, GAT, like that, for different 5 or 6 nucleotide combinations, the current will be generated. Then using a hidden Markov model, the current will be used for base calling. So if you see the sequencing machine, this is only a 90-gram small sequencing machine. You can connect to the computer using a USB 3 port. But the sequence read what you get will be more than 1 MB base. Up to 1 MB you may get it. And suppose when you do a Sanger's method for reading the sequence after doing the PCR, also after doing the sequencing PCR, then after loading into the machine, it takes almost 2 hours to do the capillary electrophoresis. Then it will take the reading. But here, the base calling is done per second. 450 base pairs are passed through the pores. So for that, the current is generated. So, and it is also a real-time sequencing. in the case of real-time sequencing means when you take a ABI Prism machine, when the sequencing is, when the fragments are passing through the capillary electrophoresis, you can't do the base calling. But in the case of nanopore, when the DNA is passing through the pores, when the current is generated, when the that raw files are accumulating, that raw files also you can use for real-time sequencing. But you need a very high bandwidth internet. So if there is an internet breakage is there, once again it will start from the beginning. So because of the internet problem, right now we are not able to do in India the real-time sequencing, but you can try for smaller database or smaller library. You can try with the real-time sequencing, and I told you the equipment is very less expensive, and there is no amplification or there is no cloning, there is no PCR, etc. in the case of nanopore sequencing. And when you do the sequencing reaction also, you can finish it everything in 60 minutes. Because now we are using only 1D method. We are not using the two-dimensional method, means double-stranded DNA sequencing. We are doing only single-stranded DNA sequencing using the nanopore because the flow cell is designed like that. And this rapid 10 is for sequencing small viral genomes. I'll tell you, there is a small flow cell is called SMUTGeome is there. In that SMUTGeome, you can sequence a single sample, a small viral DNA or small bacterial DNA. You can sequence using SMUTGeome. So that SMUTGeome flow cell you can connect to a iPhone, and in that the sequencing reaction will take only for 10 minutes. Then that 10 minutes reaction mixture you can load into the flow cell, and in the field, if you are going to a field, agricultural field, directly you can sequence the sample there itself. 10 minutes at a room temperature you can do it. So this is actually the items within the Minia. So this is the sequencer. So here the circuits for collecting the currents, everything is there. And this is the flow cell, which is the consumable. So this is, so even though the equipment, everything is very low in price, but this consumable is a little bit costly because this flow cell, this having a biological membrane, so we can't reuse it several times. We can wash this flow cell once and we can try it, but how much data we get it in the first use like that, we may not get it in the second time. So this is a consumable flow cell. So the pores are actually at this location. You can see a yellow colored portion. So the pores are arranged there, and you have a sample port here. So the DNA library will be loading at this sample port, and through the sample port, the samples will be coming here, and you can connect this. So there is a port is there, so you can connect it to there. So this is the port for connecting the USB to the computer, and there are circuit is there to collect the electric current what is coming from this flow cell. So the workflow for the nanopore sequencing, there is like a DNA extraction. So here in the case of next generation sequencing method, we always prefer high quality DNA. Very high molecular weight, high quality DNA without any, the 260, 280 by ratio. So we need a very high quality DNA without much shear. So generally we use a cubic machine to measure the quantity and quality of the DNA. And next step is library preparation, then sequencing using the Minia. Then whatever data we get it, that will be used for base calling. So in the case of Minia, suppose if you are loading 750 nanogram of library into the flow cell, you have to sequence for 48 hours so that your entire sample will be sequenced. And from 2 minutes, you will get data. Suppose if you are using a control DNA like a lambda phage, and if you have included lambda phage into this library, after 2 minutes, if you do a base calling, you can see a lambda DNA fully sequenced. So that is the advantage. You know the size of lambda, around 47 kb. So, it is impossible to get a 47 kb sequence in 2 minutes in a AVI machine. So this is the advantage. So in a short time, a longer read will be sequenced in the nanopore. So this rapid method I already told you. This is you can use it for field purpose, and the 60 minutes method and all used for multiplexing when you have more number of samples. So these are two methods. So this is transposase based method. That is the one which is generally used in the SMUD-Geon sequencing. You can these reagents are very stable at room temperature. So you can take it to the reagents. You can take it to the field, and you can isolate the DNA from the sample, what is leaf virus or whatever it is, or bacteria, whatever you isolate from it. You can isolate the DNA and you can mix with this transposase based sequencing reaction mix, and you can load into the SMUD-Geon. You will get the sequence. So these two steps are for actually when you are not doing multiplexing, single sample. You are not adding any barcode for identification of different sample. Directly you can do a preparation for ligating the adapters. Adapters includes the motor proteins and the Y adapters for recognizing the end for directing the DNA through the pores. So this step is actually for when you do handle a single sample. And when you are doing a multiple samples, it's so these two this method you cannot do it in Minion, because Minion flow cell minimum you have to use 750 nanogram of library DNA. So this is the method suit for SMUD-Geon only, single samples. But there is also one flow cell is called a Flongle. So there also the number of pores are less. So your chance of getting DNA sequenced is more there. It's more compact. Pores are more compact, so you can use there. So in the Minion, suppose if you are doing a soil microbiome, you can use up to 30 samples. For 30 different samples you can use. So when you use that samples, you have to distinguish the different samples from different locations. So each samples you can label with the different barcodes. So after end preparation, you are ligating the samples with the different barcodes. So you are when you are doing the multiplexing, you can differentiate your sample based on the barcodes. When you do the base calling, a different folder will be made in the computer depending on the barcode 1, 2, 3 like that. So if your sample 1 is labeled with the barcode number 1, the DNA sequence of sample number 1 will be stored in the folder barcode 1. So like that, you can use up to, there is a kit available up to 96 barcodes. And these 96 barcodes you can, suppose when you are doing a PCR fragments for sequencing, you need to generate only very small amount of DNA database only. So in that case, one Minion can hold 96 samples. So 96 different barcodes you can label it for different samples. Like after barcoding, you are ligating with the sequencing adapters. So I told you the sequencing adapters you are putting to direct the DNA through the pores, and also to the motor proteins to attach to do the helicase unwinding of the strand, so that it will pass through the pores. So and there will be also a tether protein will be there to attach the DNA to the pores. After that only the helicase protein will become active and it will pass through the pores. And suppose if you have a very less amount of samples, so suppose if you are isolating a DNA from soil, and if you see the population of that particular bacteria or flora is very less in number, then in that case you can use a PCR method. So there what you are doing is the PCR primers you have to make in such a way that it will hybridize with the adapters. Then you are doing a PCR. Then so that time when you do the PCR, through the PCR the adapter will be ligated with the template, so that you don't have to do a ligation reaction to attach that adapter to the template. Then that adapter ligated templates you can use for the addition of the helicase protein and tether protein. Then you can load it into the menu. So this is actually how a pore is looking like. So this region is the MSPA is loaded. So like this, 4 channels, you can see it here. So there are, suppose when you see the flow cell of Minia, there will be 512 channels will be there. In each channel there will be 4 pores. And this among the 4 pores, one pore will be actively sequencing the DNA, and all the pores at a time it will not be sequencing it. So first what we'll do, when we attach the flow cell to the computer and when we run the flow cell using Minnow program, the program will check all these pores and it will arrange in such a way that the active pores, highly active pores will be first used. So like that, in that also they will categorize like 4 sections. That is called max scanning. So in that max scanning, 1, 2, 3, 4 will be there based on the ability of these pores to sequence the DNA. So then every 4 minutes, again one max scanning will be done. So sometime what will happen when a long fragment is passing through the pores, in the middle of it, it may stop. It may not pass through it. So because of that, some pores will become inactive, or because of the temperature fluctuation, or because it's a biological membrane protein, anytime it can lose the kinetic activity. So because of that, some pores may become inactive. So because of that, every 4 minutes, it will be doing a mass scanning, and it will see which pore is highly active. Then those pores will be used for sequencing the DNA. So in 512 channels is there. In each channel there are 4 pores are there. So when you purchase a flow cell, that is Minia flow cell, there will be 2048 pores will be there. So when it is transported and when sometime when we get through the customs, we should need all 2048 pores active so that we can use it for the full capacity of the flow cell. So generally you can store the flow cell for 1 month at room temperature, but it is always advisable to keep the flow cell at 4 degrees centigrade so that the pores will be active for longer time. And after once using, there is a wash kit is available from Oxford Nanopore. There is nuclease, everything is there. After sequencing is completed, you can wash this flow cell. There is a protocol is there. And you can remove entire sample what you have loaded in the flow cell. Then you can again recharge with the electrolytes and you can store it in 4 degrees. Then before using the second time, you take it and do a sequencing run and do the base calling. And you see whether any DNA you are getting from the previous load. Suppose based on that, how much DNA you are getting from the previous load, either you can use the same barcode or different barcodes. But it is always advisable. Suppose if you have for the first run, if you have used the barcode of 1 to 12, next time, second time you use barcode 13 to 24. And you can see what is the sequence length obtained. Around 11 MB sequence also obtained from the published data. So these are actually the parameters for checking the efficiency of the pores when it is doing the sequencing. So in the real time, you can even see what is the larger fragment sequenced. So I obtained once 21 EMD sequence. So this is the result what you get after sequencing. So the sequencing is when it is going through the nanopores. The files will be stored in the FASTA file format, and this FASTA file format will be used for the base calling. So after base calling, we get to the FASTQ reads. So here all parameters through the first you will be having the DNA sequence. After that, at what time, through which pore, how many minutes the DNA sequence. So all those details are there in this script, what you are seeing in the below. So generally, if FASTQ reads, you have to keep it safely. Suppose when you are doing the sequence analysis for a nanopores. Suppose when you do any change in this, further sequence analysis will become very difficult. So you have to always keep these FASTQ reads very safely without removing any of these asterisks or comma or full stop or anything like that. Suppose if the FASTQ reads you want to convert to FASTA reads, you have to say this is a sequence generated from a nanopore to convert to FASTA reads. So if anything is missing in this script, then it won't convert it. It will show error. So because of that, after FASTA reads, the FASTA reads you won't see any sequence. These are all raw data, and this FAST file we will be using for base calling. So so for base calling, there are I said there is a real-time base calling is there. You can connect to the nanopore server and you can do real-time base calling. Otherwise, because of the internet problem, we can do the base calling in our system itself, computer itself. Earlier, the system
Ma'am, it is not audible.
Ma'am, he has left the meeting. അല്ലല്ല മറ്റേ നടന്നുകൊണ്ടിരിക്കുക ഇയാളുടെ ഉണ്ട് RIPD ചോദിച്ചാ പറയാൻ ചോദിച്ചില്ല അല്ലെങ്കിൽ ചെയ്തോട്ടെ ചോദിച്ചു സിബിളിന്റെ ഇതിപ്പോ എന്ത് ചെയ്യും അതാണ് Something happened to the connection. അല്ല കട്ട് ചെയ്യുന്നതല്ല, നമ്മൾ എടുക്കുന്നില്ല.